The speaker is Ainara Elorza Peregrina. She's going on the CBM, CSIC, and her talk is titled RNA Sequencing Reveals Alternative Splicing Deregulation in Striatum of Huntington Disease.
Okay. Good morning. First of all, I would like to thank for inviting us to present our work here today. I work in José Lucas' lab at the Centro de Biología Molecular, Severo Ochoa, in Madrid. Our work wants to explore whether there is a global alteration in alternative splicing in Huntington's disease striatum. As you know, striatum is the main affected region in this disease that is caused by a CAG. That is caused by a CAG repeat expansion in exon 1 of Huntington gene. Symptoms appear when the number of CAG repeats is higher than 40, and this CAG repetition encodes for a polyglutamine tract, an expanded polyglutamine tract in the N-terminal region of the Huntington that aggregates, it's responsible for the aggregation of these aberrant proteins in the characteristic inclusion bodies that are formed inside the neurons in the striatum. There are also other neurological diseases, such as some spinocerebellar ataxias, where the CAG repeat expansion is also in the coding region of the different affected genes. And in all these cases, this CAG repetition results in aberrant proteins with this polyglutamine expanding tract. But there are also other trinucleotide disorders where the CAG or the CUG repeat expansion is not in the coding region of the gene. So in these cases, it's not so obvious to think in the polyglutamine as the cause of the toxicity. And in fact, in some of these cases, it's been described that the RNA itself has a toxicity through an alteration of the alternative splicing in these neurons. The best characterized case is the myotonic dystrophy 1, in which the CUG repeat expansion is able to conform a stable structure that sequesters or alters the activity of several splicing factors, resulting in a disruption in the splicing of the genes that depend on these splicing factors. So, what is known about splicing alteration and Huntington disease? There are some reports already in the literature. The first one, some years ago, described that the CAG repeat expansion in the Huntington messenger disrupts its own splicing, generating N-terminal and highly toxic peptides of the Huntington protein. And in this work, they also suggested that the splicing factor SRSF6 could be the responsible of this alteration, because through a bioinformatic prediction, they found that its binding site matched almost perfectly with the CAG repeat expansion present in the mutant Huntington. Some years later, another work, this by our lab, in fact discovered or described that this SRSF6 splicing factor interacts with the mutant Huntington in the inclusion bodies inside the neurons, and that its activity and mobility is completely altered in the neurons of the Huntington disease patients. And this alteration correlates with an imbalance in the ratio of the two main splicing isoforms of the tau gene, the 4R and the 3R isoforms, and it was well known that this alteration in the balance of these isoforms is enough to cause neurodegeneration in an inherited form of dementia. And in our work, we could also demonstrate that it contributes to the pathogenesis in Huntington disease. A third work also by our lab last year, demonstrate a third splicing alteration due to this SRSF6 splicing factor, in this case the MAP2 gene, that might be contributing to the degeneration in the dendrites described in Huntington's disease. So we have at least 3 SRSF6 targets which splicing is completely altered in Huntington's disease. And at this point, we reason in the lab that probably many other splicing or genes with alternative splicing might be altered in this pathology and might be contributing to the neurodegeneration. So we thought that the first way to study them could be, and the unbiased way, could be doing an genome-wide analysis of massive sequencing of RNA and mRNA. And we decided to do it to focus on the most relevant and affected region in the disease, the striatum. So we took 3 samples from controls and 3 samples from HD patients. And to avoid the noise associated to the neuronal loss, we decided to make a comparative RNA-seq with samples coming from mice, striatum samples from 3 controls and 3 R61 mice, that it's a transgenic model of Huntington disease. So here I show you, well, the first analysis. We could find that more than 5,000 of significant differentially expressed genes were altered in the human samples and almost 2,000 in the samples of mice. And here I show you just a quality control of the RNA-seq, where you can find, or you can see that the samples group properly, keeping the 2 biological groups separated in both cases, in both species, human and mice. And then we did the analysis of the alternative splicing. We used 3 bioinformatical tools, RMATS, BASTools, and San Juan. And we could find a high number of genes with their alternative splicing altered. In all the 3 analyses, you can see in red the human genes with these alterations. This number, in all the cases, as I said, is much higher than in mice, as expected. The diagrams in purple are the genes of the mice samples. But the genes that were interesting for us were, as I mentioned before, those that have an alteration both in human and in mice, in both species. So, taking into account the 3 intersections of the 3 analyses, we could have 344 genes with a dysregulation in their alternative splicing. And that this data allow us to say that there is an striatal signature of missplicing in this disease, but is it contributing to the pathogenesis? We make an analysis with the Ingenuity Path software, and we found that we had a clear enrichment in genes related to neurodegenerative or neuromuscular disorders. And moreover, several of these genes are directly involved in a monogenic neurodegenerative disorder. So at present in the lab, we are validating this alteration at biochemical level, and we are checking if this alteration in their splicing has an impact on their protein levels, on their functionality, or on their subcellular localization. This would indicate, in case of being in that way, that would indicate that probably they are contributing to the neurodegeneration. So the other thing that have interest for us was to determine if the alterations in migration and phosphorylation observed in SRSF6 splicing factor mirrors an increase or a diminished activity. To answer this question, we generate conditional transgenic mice in which the exogenous expression of SRSF6 is controlled by a minimal promoter that is only activated in the presence of a transactivator when we combine these two lines of transgenic mice. So this allows us to express SRSF6 specifically in the regions of our interest, striatum, cortex, and hippocampus, since the transactivator is under the control of a CAM-kinase2 promoter that is only expressed in 4 brain regions. When we combine these two lines, we have obtained very recently a couple of transgenic lines with a very good expression. A very good expression, as you can see, of the beta-gal reporter gene that is also under the control of the same promoter as our exogenous protein, and also the levels of mRNA of our exogenous protein, as you can see in both lines, has reached very good levels. So with these mice, now we are trying to determine if the overexpression of SRSF6, this gain of function, gives a similar or a complementary splicing pattern in the mice, and when we combine these mice with RCS1, if we are exacerbating or rescuing his missplicing pattern, and if we are able to increase or rescue the phenotype of RCS1 mice. So in summary, we have demonstrated that there is a global alternative splicing alteration in HD striatum, that the genes that with an altered splicing show a clear enrichment in neurodegenerative and neuromuscular disease-related genes that might suggest that could be contributing to HD pathogenesis, and that SRSF6 overexpression transgenic mice generated, we hope that will help us to explore whether SRSF6 gain of function mimics or counteracts RCS1 missplicing pattern. Finally, I would like to thank all the team in the lab and our collaborators. And if you have any questions, thank you.
Thanks for the talk. So my question is that for the first time, the GWAY A analysis, if you also perform in the cortex or in the hippocampus, just to know how specific is these splicing variations in other brain regions in HD?
Sorry, I didn't hear you very well. If we have performed
all the RNA-seq analysis in other brain regions besides the striatum, cortex, or hippocampus, to know how specific is the...
No, we have focused only in striatum because we think it's the most relevant region, but there are already some publications, a couple of them, that have analyzed the cortex of HD patients. And of course, we have made a comparative analysis with our RNA-seq, and well, there is some overlap, but not too much, to be clear. So yeah, I could say that we have a specific striatal signature in this case, just comparing to cortex, because at my knowledge there is no data in hippocampus, as far as I know.